Jai Hind everyone, I am Pankaj Kumar and today's video is very important. In today's video, we are going to read about the DNA fingerprinting, or you can call it as a DNA profiling. So, or you can say DNA fingerprinting. So, this is a technique by which we can compare or we can mark our segment of the DNA, or we can mark a particular gene or particular site in a DNA. So, what is the What is the main motto of this for making a DNA profiling? So DNA profile, like I am a Pankaj Kumar. Okay, so if I am a Pankaj Kumar, so what will be my profile? My profile will be like what is my sex, male, address, suppose Buxer. Okay, and then state, Bihar, and uh, then what? And then father name. Okay, father name something. Father name HK Beta, like this. This is my profile. And another profile, suppose this is the Shishma profile, so sex, female, address, Jharkhand, state Jharkhand, district Rampur. Okay, and then father's name Chandan. Another more, one more profile I am telling you. This is a Ved. Okay, so Ved, it is sex, male, and address Buxar, Bihar. Father's name, father's name Pankaj. Okay, so suppose if you'll see the similarity between the Pankaj, Susma, and Ved, among all these, who are more similar? So your answer will be like Pankaj and Ved, because we both are male, we both live in the Buxar, our states is same, father's name will be different. Okay, so more similarity will be what? This Pankaj and Ved. But if you'll see Susma, this is completely different. So for that we are making a profile for comparing the DNAs of a two individual, of a two species, or whosoever you want to do. So you understood this. Pankaj and Ved are more similar. Like that, if something will be happen, then if you want to come to the if you want to come to meet Pankaj, then there also you can meet the Ved, because they have a more similarity. Their address is same. So this is the purpose. This is the idea, rough idea, that how what is the need of the profile. Now, if I'm saying a DNA profile, so take a DNA profile of PKJ, Pankaj, Sushma, Rakesh. So DNA, what we will do? We will do the RFLP. Okay. What is this RFLP? So RFLP is a restriction fragment length polymorphism. Okay. So in this, what we will do? We will make a DNA fragments. Then we will run a band, run a gel, and then after we will get a bands like this. I will tell you about these bands. In Susma, it got like this. Suppose in the case, it got like this. Okay. So if you'll see, the Rakesh and Pankaj similar bands are more similar because they are giving the same length, but it's not exactly same because we have a different genes. At least some different bands will be there because of some different repetitions. Okay, so these are what similarities. So you can say the PKJ is more closer to the Rakesh because their bands are very similar. So in closer in what sense? It can be they can be brother, they can be uh, siblings, like they can be other relatives. So we can see the more matches. If it will be 100% same, then what you can say? Is it possible to make a 100% same? It can be possible if the two persons are monozygotic twins. Okay. In case of monozygotic twins, in case of monozygotic twins, the bands will be 100% similar, and in other different cases, you can see it can be different. Okay, bands will be different. So this is the just a profiling of a any two individuals or any other organism. Now, beta G, please focus there. DNA profiling you understood. Hope so you understood. If you have any doubt, you can send your doubts in U Biology app. Okay, or you can ask in the YouTube comments. It is always open. Got it? In U Biology, we are giving the test series for the GAT-B, CUT-PG, NEET examination, CSIR. So you can go and watch it there and have a look of that test series. That is a free, so you can have it. Now, let's continue with the discussion. So DNA fingerprinting was first discovered by Sir Alec Jeffrey. Okay, Sir Alec Jeffrey. He discovered this technique, ac- this DNA profiling in University of Leicester in England. Okay, Leicester in England. So proper definition, you can write what is the proper definition of this DNA fingerprinting. So the proper definition of the DNA fingerprinting is it is a technique which used to distinguish the two individuals of the same species. Okay, so you can write it is a technique. It is a technique to distinguish with the technique to distinguish uh, between two individual individual of same species same species in terms of in terms of in terms of what in terms of mm, in terms of DNA. Okay, in terms of DNA fragments or variant. So DNA have the several regions. So we'll understand little bit about the DNA. So in eukaryotes, we have a repeated DNA. Okay. In prokaryote, there is no repetition of the DNA. Eukaryote have repeated sequence. And this repeated sequence will be of different type, microsatellites or mini satellites. That repetition will be formed, and it depends upon the two types: micro, okay, micro, microsatellite length will be around 15 base pairs, and it will be repeated around 25 times, 20 to 30 times. And mini satellite, if you'll see, mini satellite, it is around it is around 250 or 20 kbs. So it will be around how much? 225 kb you can write, similar or equal to 25 kb, 20 kb, and it will repeat around 25 to 30, 25 to 30 repeats. Okay, here it will be 10 to 15, 10 to 15 times it will be repeating. 10 to 15 repeats. Okay, make it like one less than 150 or 15 is also correct. You can write. So this is the micro satellite and the mini satellite. So mainly in this repetition will called as a VNTR. Like this is a long DNA. Okay, suppose this is a long DNA, and this blue color is a this blue color is a micro mini satellite. Okay, so you will find this blue color, if it is a mini satellite, this is more repeated in so many times in a sequence. Okay, it will not repeat only in one strand, it can repeat in the both strand. So it will repeat mini satellite will repeat 25 to 30 times in a uh, same locus of a DNA. Okay, so this repeats will called as what? This repeats will called as a VNTR. Majorly, if you'll see in DNA profiling, you will find that will be often the mini satellite will be often used to loosely mean to the it will be often used for meaning of the VNTR. Okay, and microsatellite you can consider it also. So this micro and mini satellite will be the repeated sequence, and in this DNA profiling, this repetition will called as a variable number of tandem repeat. What it is called? Variable number of tandem repeats. Okay, so the what does it mean? The tandem, some sequence of fragment kind of things which is repeating tandem repeatedly, and this same repetition will be like we can see in the restriction fragment length polymorphism. Okay, and once you will run this gel after digestion of this repetition with the restriction enzymes, once you will run these gels, there will be several type of repetition you can see here, and according to this repetition you can find the you can solve the cases of the daughter and the parental cases or the criminal cases or the uh, more related species if you want to find. Okay, so that things you can do. Got it? This is called as a DNA profiling, and hope so you enjoyed this video. If you are liking this video, please uh, watch complete videos and go and download the ubiology app. Up to here watching till now. Thanks for watching. We'll meet in next video.